So my name is Syama Chatterton. I'm the pulse crop pathologist out of Ag Canada in Lethbridge. And Ken asked me here today to speak about some pulse crop diseases and also a little bit on some work we've been doing with spore sampling. But I'm guessing here, as no one is from the Bow Island area, does anyone grow dry beans? I thought maybe we'd get some dry bean growers out this area. So my spore sampling project might not be that relevant to you, but I still think it's interesting. But I'm going to start with talking about some of our diseases of peas. And I've kind of learned that Ken never has diseases in his pea crops. I don't know how he does it. So I've started bringing my own wherever I go. And the peas are actually looking pretty good right now. These are start from some of our field trials. And really what I primarily work on is root rot of peas. And I'm sure people in the past, if you've been to Agronomy Update or I think some of the Farming Smarter conferences, you've probably heard me talk about the whole pea root rot issue. And so what I brought here with me today is two examples of our kind of two very different pathogens that cause root rot. Very lucky this year that they seem to be very isolated. I've got some really nice Fusarium samples and some really nice Aphanomyces samples. Usually the two kind of go together and it's a big mess and you can't tell which is which. So I'll start with what I call my D-list pathogens, which is Fusarium. So we find Fusarium species pretty much in every pea crop that we go into and survey. We're almost always finding Fusarium. And I call it my D-list pathogen because it is everywhere. We don't always know what it's doing, and so I kind of liken it to something like the Kardashians. I'm sure everyone is familiar with them. They're kind of famous. I don't know why. I can't quite figure out what they're contributing to society. And that might be the same with our Fusarium pathogens here. We find them everywhere, but yet we can get plants that still look like this. Now this is under irrigated production in one of our trials, so they're not really suffering from drought stress. But when you look at the roots closely, you can see that the roots are really not looking that great. So it's kind of surprising that when you have a root system that looks like this, that we can still get pea plants that are kind of waist high on me. And I'll pass these around too so people can have a better look. But first I'll just give you some things to look for. When you look at the taproot, you'll see, these are all bunched together, but I'll start passing them around so people can have a look at what I'm talking about. I'll just keep one for myself. Okay, here we go. Let's see, yeah. So with Fusarium root rot, when you look at the taproot, what you often will see is you get a kind of a black lesion just along the whole taproot. The lateral roots still look fairly healthy, and we can still see some nice nodules on those roots. I didn't bring a knife with me, but oftentimes if you kind of just try to crack it open, like this one, you crack it open and it's really nice, and you can just use your fingernail even to kind of peel out that outer layer, and you'll see that there's a nice red vascular kind of streaking inside of the vascular system. And that's because the Fusarium gets into the vascular system and that's why it starts causing damage to the plant. So that's kind of our first pathogen. And then the other one that I call my A-list pathogen is Aphanomyces. And this one is a very new player on the scene. So we only found it in Alberta in about 2013, 2014 was when we first detected it. In 2014, we had a lot of pea crops that really suffered, especially it was a very wet year that year. And we started looking for it, thinking it's a new pathogen, and lo and behold, it was pretty much present. We could find it anywhere where peas had a history of being grown within a field. And so this one gets a lot more attention because it's newer on the scene, and because we can get plants that look like this. Again, when you look at these root systems, I think it's completely shocking that the peas are actually doing as well as they're doing. They have some pods that are starting to form. Whether those will actually be able to fill out and set seed completely, I think remains to be seen. And I'm hoping I'm not now spreading Aphanomyces all over this nice field here. But I'll pass these around so you can have a look at that as well. It doesn't spread that easily.
Sure.
Okay, so I think the first thing that you can note when you look at these is that the whole root system really looks like it is, here, pass a few more here. The whole root system looks brown, and the lateral roots, there's really not much left to them. And if you feel the lateral roots, they feel really wiry, and that's because all of the outer cortex of the root has pretty much been sloughed off by the pathogen. So all that gets left in this case is the vascular system, but there's no outer material in that roots to help with any water absorption or any nutrient absorption. And then you'll see on here as well that we have no nodules left on these roots. So Nevin really gave some nice examples of why, talked about why nodules are so important and how we can rate our nodules. And then you look at something like this and you see that there's really no nodules left. And that's why I wonder how, well, these ones are particularly suffering, but you kind of wonder how that plant is still kind of able to push through and produce some of those flowers and pods that we're seeing on some of the plants. So this is Aphanomyces. It's caused by an oomycete pathogen. An oomycete is heavily dependent on water for part of its life cycle. So it leaves behind all that root material that's no longer there. Those are filled with resting spores called oospores that as that root material gets sloughed off into the soil, that's what gets left behind. And then the next time you grow peas or lentils are also susceptible, and you have a lot of moisture present in the soil, those oospores sense that there is a good host for them. They start to germinate. They produce little zoospores that then can swim kind of short distances within those water films to find the roots. So that's why this is a pathogen that is really heavily dependent on water. So why are we still seeing it being a problem in dry years? And that's because we had such a wet spring. So we had that nice wet spring to have a lot of moisture in the soil. Now we go to our field trials and they're dry as bone. There's really no moisture left. But we had early infection when there was a lot of water there, and that's why the peas look like this. But because it's dry now, the infection process has stopped, and that's why the peas are still kind of holding on a bit and will probably give us a yield at the end. The Fusarium, part of the reason why it looks so good is because, as I said, that is grown under irrigation, so we are offsetting some of the issues with the dry conditions and not being able to uptake enough water with such a root system that has pretty much decayed away. And then in terms of management, unfortunately with these soil-borne pathogens, we don't really have any in-crop options for them. There's no fungicides that can be sprayed on a foliar material that then get translocated down to the root material. Most fungicides don't have very little systemic ability, and if they're going to move at all within the plant, they usually move up towards a growing point, so nothing's going to move down into the roots. So unfortunately, if you see that you have a problem in your field, there's not really much that you can do at that point. It's just kind of serves as a lesson that the next time you grow peas or lentils, considering growing peas and lentils in that field, you might have to stop and think about the choices of the pulse crops that you want to grow. We have been doing some trials with seed treatments. There is one seed treatment, ethoboxam, that is registered for early season suppression of root rot, and that's basically what we've seen in the field. We do get an early season suppression, but this is a disease, particularly Aphanomyces, is a disease that you don't just need protection at the beginning of the season, and no seed treatment kind of has a long-lasting effect beyond kind of that seedling stage. So we might see the early season suppression, but we don't see it carry forward to later in the season. So really our most important management tool is knowing your field and knowing that if you've had a problem the last time you planted peas or lentils in the field, you probably want to stay away from growing peas and lentils in that field again, at least for kind of extend your rotations out. And we're doing some research to look at how far you might have to extend those rotations out, and also looking at whether there's other pulse crop options like faba beans. Chickpeas and soybeans are fairly resistant to Aphanomyces. They're not necessarily resistant to Fusarium, but we're trying to see how growing those kind of non-host pulse crops affect the inoculum potential in the soil. Yeah, and then in terms of, I guess those are kind of the big ones are really the root rot, and I would say that 60% of my research program is focused on studying these root rots, particularly Aphanomyces, because it's such a new player on the scene for us. We have to learn a lot about its biology and how it's behaving to be able to come up with management options. And then I think Nevin already mentioned the other big one, which is Mycosphaerella blight. And again, I thought in our irrigated trial, which looks so nice, that I maybe have some examples of Mycosphaerella, but it really just hasn't shown up yet, even under irrigation, and it's just because it's such dry conditions that the pathogen doesn't like those conditions. So I can move on to talk about our spore sampling, or maybe I can take questions on root rot first, and then I can move on to the next portion. So if anyone has questions about any diseases of pulse crops, I'm happy to answer.
Would that plant eventually totally die?
Yes, we have seen in the past that the plants will completely collapse. Under wet conditions, like we had in, say, 2014 and 2016, we saw a lot more peas collapse, whereas in the dry conditions, they still get that disease early on, but then I think the infection stops and they can still kind of limp along. If it got really wet now, then these plants, we'd probably see them go down pretty quickly.
I noticed it last year in lentils, just the odd random plant
Yeah.
was dead.
Yeah, yeah, and you see, so that's something to keep an eye on then in that field as you continue to the next time you grow lentils, because you'll probably start to see a small patch move out to a big patch, and then the next time your patches will get bigger, and then eventually we've seen fields where the whole field has been affected. And those fields are usually ones that have had maybe a history of growing peas or lentils for 20 to 25 years. So you probably had a crop in there, every 4 years. So you've had a crop in there every 5 or 6 times by the time you get to that point. But, yeah.
Does tillage break the cycle at all?
I don't think so. That's been hard to study because everything here is no-tilled. But this disease is a big problem in France as well, and they still are practicing all their tillage, and we still see it as a problem. And we've done some research trying to look at where kind of these spores and the Fusarium inoculum is kind of concentrated in the soil profile. And we do see that it's kind of in the top 10 to 15 centimeters is where it's concentrated, but we can still find it down to like the 20, 40 to 60 centimeter range. So just enough, the soil, the water percolation is enough to just kind of move it through the whole soil zone. Yeah.
How do you tell what it is?
That's a good question. Yeah, send it away. So like I said, this is kind of perfect conditions this year where I was very easily be able to see, okay, this is Aphanomyces and this other one is Fusarium. Oftentimes, Fusarium will come in on top of Aphanomyces and then you can't really tell what you're dealing with. So we do recommend that you can get your roots tested from one of the seed testing companies like BioVision or 2020 in Alberta, and there's the other places in Saskatchewan as well that provide either a tissue test or a soil test for Aphanomyces. I don't think they're doing testing for Fusarium species yet because those are there's so many Fusarium species that they can be very hard to distinguish one from the other, but they are doing Aphanomyces testing.
Correct me if I'm wrong, but if you're suspecting it and you're sending it to the lab, make sure they have PCR for doing it.
Yes, that's right.
Because if you played at the Fusarium, just eat Aphanomyces,
that's
and they won't actually...
Yeah, that's exactly correct, and that's partly why I'd say Aphanomyces is a new player on the scene, and so we expected that maybe we would just see it in 1 or 2 fields. But part of the issue has been that when people have been dealing with root rots for maybe the past 20 years, they've taken a root sample, plated it in the lab, and you're right, Fusarium often is overgrowing the Aphanomyces. It's much harder to get out of the roots, and so we need to go for a PCR to detect it. And that's really how we discovered that, okay, it is present in a lot of our pea fields across Alberta, Saskatchewan, and Manitoba. Okay.
Is it detectable at the seed stage?
No, and it's not a seed-borne pathogen. It is a soil-borne pathogen, and like Fusarium is a little more complicated. It kind of has 2 phases. So Fusarium can cause a seedling blight, and that's what a seed treatment protects against, is that seedling blight. But then the Fusarium root rot comes on later in the season, and you don't get any effect again of the seed treatment. Aphanomyces doesn't really cause a seedling blight. It comes on a little bit later, maybe when the plants are 3 to 4 weeks, provided there's enough moisture, and that's maybe why we don't see that the seed treatments provide kind of a good control, because it's not really a seedling issue. It's more of a advanced plant issue. Yeah, and I should mention that it is spread primarily. It's a soil-borne pathogen. It is spread primarily by soil. So we do talk about taking all those sanitary measures to try and prevent spread of soil from field to field. But like I said, I don't want to spread Aphanomyces here. Little bits of roots that fall here won't really cause a problem. We've been trying to create an inoculated field nursery in Lethbridge so we can start screening maybe some breeding material, and it has been very difficult to get the pathogen to actually establish and take hold. So I think they're kind of thinking about, okay, well, how is this actually spreading? How much soil would you need to move in order to kind of infect a field? And it seems like it's actually going to be a huge amount of soil that we will have to move. So there's some questions then of why has it kind of popped up in all these fields seemingly suddenly, but we have scientists have a lot of hypotheses about that, and I don't know which one is correct yet, but yeah. Okay, so that's kind of, and I guess just so we don't leave any disease material here in this field, if people can just bring the samples back up here. And then I'll just talk briefly about our spore sampling project as well over here. So what we've been trying to do, or what we started last year, and we're working on this for the next 4 years, is seeing if we can use these spore samplers to try and function as an early disease warning system for different pathogens. And because I'm a pulse pathologist, my pathogen of focus is Sclerotinia on dry bean. But it's the same, Sclerotinia is the same disease that causes Sclerotinia stem rot on canola. It causes white mold in dry bean. It causes white mold in potatoes. Basically any broadleaf crop it can infect and cause disease. So we're kind of focusing on dry bean, but keeping our eyes to the future, thinking, well, we can probably apply this to a lot of different crops. And so what we have done is put these spore samplers out in some dry bean fields. And we have 2 heights. So this one is to try and look what's going on below the canopy. And then we have some that are about this high to see what's going on above the canopy. Because one of our big questions with Sclerotinia, and I'm sure everyone's familiar with it in terms of canola, is that you get the little structure that's in the soil that produces a little mushroom-like apothecia that then release spores. So the question is all your inoculum just coming from down below in your field, or is there inoculum blowing in from a neighbor's field? So we kind of have 2 different heights within the canopy to try and see where that inoculum is coming from. If it turns out that all the inoculum is coming from your own field, then maybe we can't kind of work towards kind of a regional wide spore forecasting network. But if we have some that's being spread by the air, then we can use that to look at kind of a regional forecasting network. The beauty of this system, and I'll take off this lid if I can. I always get stuck. Okay, so how this system works is it's an active sampling mechanism. So we usually have it attached up to a solar panel, which charges the battery, and then it just runs for 24 hours a day, 7 days a week. And so it has a little fan that runs, and it pulls in air through this little air intake, and then deposits them down here. There's usually some vials here, and then they can move every day. They move, so you can collect kind of a week's worth of samples in one system. But the beauty of the system, of course, is that it's collecting everything that's in the air. So my focus might be Sclerotinia, particularly as it pertains to white mold of dry bean, but we're catching everything that's coming from the air and getting deposited into those vials. So we're hoping kind of for the next phase of the research that we can look at developing kind of a multi-crop, multi-pathogen network, so we can maybe start detecting, and I'm working with some of the cereal pathologists to see if we can start detecting things like rust, Fusarium head blight on top of our white mold. I think potato growers are interested in Phytophthora. They have a kind of a big spore monitoring network going on for that already. So my research focus is white mold, but we're going to start looking at whether we can start detecting kind of multiple pathogens within these spore samplers and see if that's relating to the disease that we're actually seeing in the field. So kind of early research phases now, but I know this is a really I guess worldwide, we're seeing a lot of interest in kind of the spore forecasting networks. I know Australia, I think, has a big network up and running. They have a network in England for forecasting sclerotinia on canola, and I think also in Poland for blackleg of canola. So I guess we're kind of hoping to start taking advantage of some of that technology too, and being able to come up with these disease forecasting networks. Even though we're just kind of in the second year of research, so it might have a long way to go, but I just wanted to take the opportunity to kind of talk about where that might be going in the future. And I think that's all that I have to say about that, if anyone has any questions.
This forecasting is going to help guys in timing and spraying.
Yes, yeah, that's the primary focus is, you know, particularly for dry beans, we really focus on spraying at a certain crop stage without necessarily knowing if there's a risk of getting the disease. And white mold can be such a huge impact on yield that producers will spray at that early flowering stage, regardless of whether there's a risk there or not. So we're hoping this will help fine-tune understanding whether there's that risk there and being able to better time fungicide use, and to just say you may not even need to apply a fungicide because maybe there is no issues at this moment. So I think it's just providing a little more knowledge on that, and that early forecasting system to know whether you need to spray a fungicide or not is really the ultimate goal. Okay.
All right, join me in thanking Syama.
Take these all off.
Yeah. So on that note too, I'm not sure, just to give a plug to the Wheat Commission, they also have a Fusarium risk management tool that's on their website, and it's also on the ACIS website. That's the Alberta Climatic Information System. So there's a huge network of weather stations across the province, probably some of the best networks around actually. And what they do is link up that sort of weather system with a risk map as well, looking at that. So I think eventually it could all evolve into that. And I think there's also quantitative type analysis that can be done down the road, just to know what your spore load is, not just whether it's there.
Yeah, exactly. And I should have mentioned that's a big thing that we're trying to do with this spore network, is hook into these sort of weather modeling networks as well, to see if the weather is a really good predictor of spore release, if we can just ultimately move to kind of a weather-based modeling network and just hook in with what's already going on with Fusarium head blight.
Send me a text message and it's
yeah,
time to spray.
Exactly, yeah.